welcome to the next educational video. So in the last video I was discussing about kingdom Monera, the classification of kingdom Monera, the characteristic features of kingdom Monera. So in this video I'm going to discuss about the bacterial cell. So let's see the ultrastructure of the bacterial cell. This is the ultrastructure of the bacterial cell, which consists of a cytoplasm, nucleoid, granular inclusion, fat globule, ribosomes, pili, flagellum, capsule, cell wall, plasma membrane, and mesosomes. In this video, we are going to study about each part of the bacterial cell. First, we will see the characteristic features of the bacterial cell. We all know that bacterial cell is a prokaryotic cell because it have a naked DNA. The DNA is not enveloped with any nuclear membrane or a membrane. So the characteristic features of the bacteria or the structure of the bacterial cell is studied with the help of an electron microscope, and internal structure of the bacterial cell was studied with the help of staining methods and with the help of some biochemical tests. Let's see the parts of the bacterial cell. The first is a capsule or a slime layer, then cell wall, the surface appendages which consist of flagellum and pili, and the protoplast which consists of a plasma membrane, cytoplasm, plasmids, episomes, and nucleoid. First we will see capsules or slime layers. So capsule is a layer present over the cell wall. So it is a gelatinous layer present on the surface of the bacterial cell wall. This capsule is made up of a polysaccharide, but in case of E. coli, the capsule is made up of a disaccharide, and also in other bacteria, it was made up of polypeptides, only 1 or 2 amino acids. In Bacillus anthracis, it was composed of D-glutamic acid. If the layer is made up of a polysaccharide, that layer is called a slime layer. If the slime layer is associated with the polypeptide chain, then it is called a capsule. Capsule present in all pathogenic bacteria. So all the pathogenic bacteria are consist of the capsules. It protects the cells from temporary drying and destruction by phagocytic cells of the host body. It protects the bacterial cell from the phagocytic cells of the host body. So that's why the capsule is present in all pathogenic bacteria. And also it protects the cell from antibacterial agents. The capsule behaves like osmotic barrier and promotes the attachment of the bacteria to the surface. It also provides surface attachment support to the bacteria. The presence and absence of the capsule is genetically determined. So not all the bacteria gets the capsule, only the bacteria which is coded genetically, then only they can able to develop a capsule. Capsulated forms are virulent, that means they causes the diseases to the animals, plants and humans, and they are known as S strains, that is smooth strains. The non-capsulated bacteria are known as a non-virulent, that does not causes any diseases to the plants, humans and animals, hence they are known as R strain, that is rough strain. Next part is the cell wall. The bacteria has thick rigid cell wall. Because of the thick rigid cell wall, it protects the internal organs of the cells from expanding and bursting. The cell wall is made up of 2 components, that is peptidoglycan and diaminopimalic acid. Peptidoglycan is also called mumeric acid. So it consists of 2 components, that is the peptide portion and a glycan portion. The peptide portion is composed of amino acid, the glycan portion is composed of a sugar. Glycan portion forms the backbone of the peptidoglycan. The peptidoglycan is composed of other alternative units such as N-acetylglucosamine and N-acetylglutamic acid joined by beta 1,4 linkages. So the gram-positive bacteria and gram-negative bacteria have different types of cell walls. You can see in the picture, this is the cell wall of the gram-positive bacteria, this is the cell wall of the gram-negative bacteria. The cell wall composition is so different in both type of the bacteria. The gram-negative bacteria have a more complex cell wall structure than the gram-positive bacteria, and also very protective, and also gram-negative bacteria have a very protective cell wall than the gram-positive bacteria. The thickness of the cell wall is ranges from 10 to 8 nanometer. The cell wall of the gram positive, as I told, the cell wall of the gram negative bacteria is much more complex. So because it has a bilayered outer membrane, so which is made up of protein, phospholipids and lipopolysaccharides. The outer membrane serves as a barrier, so it prevents the outside chemical entry inside the cells and also prevents the escape of enzymes from the cell. Next are the surface appendages, which consist of two parts, that is flagella and pili. Let's see about the flagella. The flagella are the fine unbranched hair-like structures present and protrude through the cell wall. They produced through the cell wall. They are responsible for the motility, movement of the bacteria. So it is composed of a flagellin protein. That's why it is called flagella. So there are different kinds of flagella based on the different kinds of condition. So if the flagella is absent, such as in case of lactobacillus, the condition is called antrikos. If the flagella is attached to the one pole, for example in Vibrio, the condition is called monotrichos. If two or more flagellas are at the one end, two or more flagellas are present at the same and the one end, such as in case of Pseudomonas, that condition is called elliptotrichos. If the flagella are at the opposite ends, then it is called amphitrikos. The condition is called amphitrikos. If the flagella is distributed on the surface of the entire cell, such as in case of bacillus, then the condition is called petriticus. So this is the different conditions of the flagella. So based on that, they are classified. This is a monotrichus. At the only one end or at the one point, the flagella is developed. So next is a cephalotrichus. You can see at the one point more than two flagellas are developed. That is called cephalotrichus. Then amphitrichus. The flagellas are present at the opposite point. So this is the opposite point. This is the opposite point. That's the condition is called epitrichus. Then petritricus. If the flagellas are present entire surface of the bacterial cell, so then it is called petritricus. Next is the Pili or Fimbriae. So these are the extremely fine halohelical filaments. So these are all present on the surface of the bacteria. They are present in both motile and non-motile bacteria. They are thin, short and more numerous than flagella. More number of flagella is observed in gram-negative bacteria such as bacillibacteria. So they are not used for the motility. So they will not help for the motility of the bacteria. It is composed of a protein called pillin. That's why it is called pili. The size ranges from 3 to 25 micrometer in length and 0.5 to 25 micrometer in dia. Next is a protoplast. So it consists of a plasma membrane, cytoplasm, nucleoid and a plasmid. First we will see plasma membrane. Plasma membrane is present under the cell wall. It lies beneath the cell wall. Plasma membrane forms a convoluted vesicles at the center of the cytoplasm. So that convoluted structure at the center of the cytoplasm is called a mesosome. Mesosome consists of enzymes that are associated with the respiration. The mesosomes are functionally similar to the mitochondria of eukaryotes. They have two types such as a central and peripheral. So if the mesosomes are deep inside the cytoplasm, they are called central. If the mesosomes are not deep inside the cytoplasm, then they are called shallow. It consists of living and non-living inclusions. The living inclusions are ribosomes. They are present in a large number, and some of the membrane-bound organelles such as mitochondria and etc. are absent in cytoplasm. The sedimentation volume of the bacterial ribosome is a 70S. Each ribosome is consist of two subunits. They are unequal, that is 30S and 50S. Subunit is composed of RNA and protein. The non-living inclusions are volutin granules, PHP, that is polybeta hydroxybutyrate, and elemental sulfur. First we will see volutin granules. So these are known as a metachromatic granules. So they appear as a electron dense bodies. So they serve as a source of phosphate. Next is a PHP. So it is a lipid-like material. It can serve as a reserve carbon and energy source. When the bacteria is in aquatic habitat, have a many gas vacuoles which provides a buoyancy to the cells. For example, this is the bacteria present in an aquatic environment. So there are lots of water in an environment. This is considered, these are the lots of water around the bacteria. So it provides the buoyancy. PHP provides the buoyancy to the cells of the bacteria. That means whatever the more amount of water present inside the cell, they are removed out of the cell to the environment. If when the bacteria are living in an aquatic condition, so the water is removed out of the cell, the excess water is removed out of the cell. Next is the nucleoid. Bacteria cells do not have a well-defined membrane-bound nucleus. That's why they belong to the group of prokaryotes. The DNA is naked. The DNA is confined at the center of the cytoplasm, so that's why it is called a nucleoid and chromatin body. Next is the plasmids. So these are the extra nuclear genetic material which is circular and found in DNA. So these are double stranded. It is first found by Zinder and Lafferberg in 1952. They demonstrated the presence of the plasmid DNA in Salmonella typhimurium. Sometimes the plasmids are present in Archaea. These are the oldest species. So they carries the gene that was helpful for the survival of the bacteria. That is, they carries the gene which is responsible for the antibiotic resistance and also against the antibacterial agents. So you can see this is the bacteria. These are the circular double-stranded plasmids. These are the genetic material. This is all about the ultrastructure of the bacterial cell. I hope you understood and thank you for watching. Hi friends, subscribe to my YouTube channel and also press a bell icon to never miss an educational video.